The first question, identify the untrue statement about speciation. Polyploidy is an example of allopatric speciation. No, it's the one really good example of sympatric speciation, because you remember that polyploidy results from the doubling of chromosomes so that the parental type, which is a diploid crossing with the tetraploid type, can't produce viable offspring. Vicariance events promote speciation. True, we introduced that as one of the major mechanisms of geographic speciation. Biological species concept emphasizes reproductive isolation. Absolutely. Under certain circumstances, natural selection can overcome gene flow and cause sympatric speciation. We talked about an example with the apple and hawthorn flies. Species can be defined as different groups of organisms that constitute evolutionarily independent lineages. Yes. Number two, phylogenetic trees are a depiction of evolutionary history. Well, that's certainly hard to get wrong. The node on a node on a phylogenetic tree is found only at the base of the tree. That's not true. There may be a node at the base of the tree, but there are nodes elsewhere where two branches diverge. That's a node. So that's the false answer. The Frentic approach to tree estimation is based on computing a statistic that summarizes overall similarity or distance among a set of populations. True, I emphasize that. Synaptomorphies, or the shared derived characters of the species under study, are a key element of the cladistic approach to tree construction. Absolutely, we talked about that and emphasized it. The term homology describes the situation where traits are similar because of common ancestry. Yes, and we looked at examples of homology versus homoplasy. Identify the false statement. The four sources of bias associated with fossil record are habitat, temporal abundance, and taxonomic. Yes, we talked about that on Wednesday. Radiometric approaches are used to provide absolute time estimates for fossil materials. Absolutely. There have been 5 major extinction events in the last 500 million years. Yes, we showed you a figure that illustrated that and talked about it. It is not possible to design experiments to explain how life might have evolved on the primitive Earth. Oh, false. We talked about how we can derive experiments, how we can actually design experiments that give us some insight into how life might have evolved from chemical principles. And finally, the atmosphere of the early Earth lacked oxygen. True. Okay, do you have the histograms? So just to see how everybody did. Okay. Question 1, yeah, good. 2. Wow, everybody did well there. Okay. Okay, so that gives you a sense of how you all performed. The quizzes are important. They count, remember, they're 19% of the total grade. You get 1% just for filling out the evaluation on the 2 instructors. 19% for the quizzes, 40% for the midterm, 40% for the final. So the quizzes count in total, add up to half as much as either the midterm or the final. So it's important that you do your best on them. So let me just begin again with a brief review of the materials we talked about last time. We talked about the time scale of life on Earth. I mentioned that the book has a really nice several-page geological time scale that you want to look at that outlines the major epochs of geological history and then associated with it the different major events in terms of the evolution of life. And then it also gives the absolute times. So it's a nice reference. We talked about the origins of life, talked about a definition of life as simply a self-sustained chemical system that's capable of undergoing Darwinian evolution. That's life at its most elemental, most simple. And then we went ahead and talked about some ideas about how life might begin. Some are scientific, some are non-scientific. We focused on the scientific explanations and said that in order to design experiments, and this goes to one of the questions on your quiz, we needed hypotheses that we needed to identify out of the universe of all the possible ideas, that small subset of ideas about the origin of life, which are consistent with the laws of chemistry and physics, okay? And then from that, we deduce hypotheses. They also have to be consistent with the law of natural selection. We know these systems have to be able to evolve after the origin of life in order to have elaborated all of the biological diversity that we see on the Earth today. So we use logical deductions from these two principles to come up with ideas about potential experiments. And we talked about one of the early experiments, the Stanley Miller spark chamber experiment, which is a nice representation of asking how the basic building blocks of life might have appeared in the very early primordial Earth. I went on and then described to you one of the contemporary hypotheses about the very earliest life on Earth, that it might have been an RNA world, because RNA has both an information transfer capability and a catalytic capability, so that it could do both things. It could drive a very primitive metabolism because of its ability to catalyze reactions, as well as transfer information to successive generations. And then we started to talk about bacteria and archaea, and that's where I will pick up now. Now, one of the things that we started to talk about in the last lecture is how those three domains of life differ. And this table, which is straight from your textbook, gives you some of the key ways that the three domains of life differ. It's a very straightforward table. One of the big important ones is a nuclear envelope. Only one of the three domains of life encloses the hereditary material, the chromosomal material, inside a nuclear envelope. That's eukaryotes. That's what the lineage that we're a part of. It's not true for archaea or for bacteria. And in fact, there's a very interesting contemporary hypothesis that the origin of eukaryotes may have actually been from an endosymbiotic association between an archaeal cell and a bacterial cell, and that ultimately many of the archaeal genes may have been donated to the parent bacterium, but the nuclear envelope is the residual of that endosymbiotic association. The evidence in support of that is not overwhelming, but it's good enough to see it as a viable hypothesis. And we will see, probably on Monday, we'll talk about another example where the evidence is absolutely overwhelming, where the organelle in eukaryotes responsible for photosynthesis, this is in plants and in algae, the chloroplast, is the result of an endosymbiotic association between a bacterium and a eukaryotic cell. So this idea is not outlandish. There's precedent for it in other elements of evolution. Circular chromosome, not for eukaryotes, yes for archaea, and yes for most bacteria. DNA associated with histone proteins. Histone proteins are proteins that are associated with the folding and coiling of the DNA in the cell. No for bacteria, yes for archaea, yes for eukaryotes. So if you go through this list, you can see that in some ways archaea do look sort of intermediate between eukaryotes and bacteria, which is what we might think if there was a primordial endosymbiotic association between an archaeal cell and a bacterial cell, which gave rise to the eukaryotic cell. Okay, so this is a good list that tells you some of the things that differentiate these three major domains of life. The other thing about eukaryotes that is not true for archaea and true in only a few specialized lines of bacteria is that they're often multicellular. And we're going to come back to this theme of multicellularity because that was also a huge transition in evolution, to go from single-celled organisms somehow to cooperative, colonial, multicellular structures that eventually became so interdependent that they couldn't exist separately. So we'll come back to that multicellularity theme a couple of times in the upcoming lectures. But why do we bother to study bacteria? Why are they interesting and important to us? Well, one very interesting fact is that each of us is a living, walking bacterial ecosystem. There are about 10 to the 13 cells that make up the human body. But we actually carry on us, either in our digestive system or on our skin, roughly 10 to the 14th bacterial and archaeal cells. We have more bacterial and archaeal cells associated with us, with our physical structure, than we have eukaryotic cells. Isn't that amazing? So each of us is this kind of living ecosystem. There are 5,000 species of bacteria and archaea that have been identified so far by classical methods. But that's just scratching the surface. The methods in the past for identifying bacteria and archaea were based on being able to culture them individually, and most cannot be cultured. So we had no real way, well, also by microscopy, but we really didn't have powerful enough resolution to see what are certainly many millions of different bacterial and archaeal species. There are about 500 different species of bacteria in the human mouth. These are different species. Not just individual cells, they're different species. There are over 400 different species of bacteria that inhabit the human gut. So we're like a forest. It's amazing. So why? So we need to study. Bacteria are really interesting because they're part of us. And they confer a lot of benefits, but they also do some bad things. Some bacteria cause disease. There's only one known case of an archaeal disease, and that's a dental disease. But some bacteria are pathogenic. They cause diseases in humans. And this is a list of some of the major bacterial lineages and the diseases that they cause. And it's getting straight out of your textbook. You can see Firmicutes, Clostridium, you get food poisoning, botulism. Well, botulism's bad news. The botulism toxin is usually fatal. So then you have tetanus, acne, boils, pneumonia from streptococcus, strep throat. For the spirocheates, you have Lyme disease and syphilis. Actinomyces, you've got things like leprosy, tuberculosis, acne. Chlamydoles, genital tract infections, protobacteria, ulcer, gonorrhea, ear infections, meningitis, food poisoning, plague. Okay, so bacteria are also very important to study and understand because they are a major cause of human disease, and we have to know how to deal and control human diseases. One of the first, the Nobel Prize in Medicine and Physiology was first awarded in 1901. In 1905, it was given to a man named Robert Koch, who played a crucial role in showing that a bacterium caused the disease anthrax. And along the way, he came up with what were called Koch's, or what are presently called Koch's postulates, which are still used today to try and establish absolute proof that a particular bacterium is responsible for a particular human disease. And there are 4 elements to Koch's postulates, and they're given here on this slide. The first is that the microbe has to be present in the sick individual. That's not too surprising. If there's a causal relationship, it better be there. The second is you got to be able to recover a sample from that individual and then show that you can culture this microbe, this organism, and isolate it in a pure culture outside of the host organism. The third step is that you then have to take a sample from that culture of the putative disease organism and infect an experimental animal and show that it causes the disease symptoms. And finally, to complete the proof, you have to be able to recover the disease organism from the infected experimental organism. If all four of those conditions are satisfied, you have complete proof of causality, that that microbe causes that disease. Now, there's a difference between correlation and causality, and one of the big problems often in science is working out causal relationships versus just observing correlations. We may start with a correlation, that is, we notice that when particular symptoms develop, they're associated with a particular kind of microorganism, but that's only the first step. We have to then go through this careful procedure to establish causality. So this is the proof of causality, Koch's postulates. Koch won the Nobel Prize in 1905 and did a lot of other important work along the way. Now, bacteria happen also to be useful in other ways. One interesting area of contemporary research is what's called bioremediation. We use bacteria to actually digest chlorinated hydrocarbons. These are ring structures which are toxic, things like dioxin and stuff, or to help us disperse and digest petroleum molecules in major oil spills. There's a lot of work on actually applied evolution in taking bacterial species which are capable of, for example, digesting petroleum products, and then selecting them to make them more and more efficient so that you can then use them to seed, that's point 2, to seed into an area of pollution, this much more efficient microbial digester of the pollutant product to actually detoxify it and get rid of it, get it out of the environment. It's a very important area of work. The other thing that's done in, again, areas like oil fuels, oil spills, is simply to fertilize the area of the polluted area. That is, you put in a limiting resource. Often the limiting resource is nitrogen or phosphorus, because there's usually not enough to accommodate larger populations. So if you fertilize the area by adding a limiting resource, you're making it possible for more microorganisms to grow and therefore to effectively digest the contaminated area. So bioremediation, it's a big area of contemporary work because more and more we're faced with problems of environmental pollution, and we need to have powerful tools for dealing with them. And this is one way that bacteria are very important to us. Now, there's another interesting feature about bacteria and archaea. Some of them are what are called extremophiles. What do you think that means? Phile comes from the Greek to love extremes. These are organisms that come from very extreme environments, either very, very high temperatures or extremely salty environments in which most contemporary organisms cannot live. In fact, there's an example in your book of, you've all heard of deep sea vents. These are areas where volcanic material, not so much material but gases, are escaping from a vent in the underocean, and the temperature of the water around the vent is very high because of these escaping high pressure, very hot gases in the area of 300 degrees centigrade or so, 3 times the temperature of boiling water. And it's been possible to isolate archaea from those areas that are existing in temperatures of at least 121 degrees centigrade, way above the boiling point. It's pretty impressive because we know that most proteins are actually deactivated by high temperature. But these guys have evolved to live in very high temperature environments. Now, why is that of use? Well, one area where it's really been powerful for those of us who like to do science and who sequence genes is that we've been able to get enzymes from these extremophiles, from high temperature-loving bacteria, and use those in experiments like what's called the polymerase chain reaction to replicate DNA, where part of that process involves raising the mixture to a high temperature to cause the two DNA strands to come apart. Well, you'd like to have an enzyme that can survive that so that when you cool it and the DNA strands reanneal, it can go to work again. And an enzyme called Taq polymerase was isolated 25 years ago or so from extremophiles. It performs just that purpose, been very powerful for scientific research. Another big feature that I've alluded to now a couple of times in my lectures about bacteria is that the whole atmosphere of the Earth was altered by bacteria, a particular kind of bacteria called cyanobacteria. Cyanobacteria are photosynthetic bacteria. You know, when life first appeared on the Earth was anoxic. There was no oxygen, no free oxygen in the atmosphere. And the energy reactions required the transfer of an electron to some sort of inorganic or organic molecule with a lower potential energy that could be used to drive biosynthetic reactions. And that went along for quite a while. And then, probably around 3 billion years ago, photosynthesis began to evolve. It turns out that you can make, under these sort of spark chamber experiments, you can actually make ring-like structures like the porphyrin rings, which are the fundamental structural molecule associated with the capture of light energy in photosynthesis. So it's not really difficult under abiotic conditions to create the molecule which is involved in light harvesting and photosynthesis. So as the inorganic materials that early life was subsisting on, which don't replenish themselves, became rarer and rarer, life was going to go away because the resources were disappearing unless there was some other way of getting a different energy source. Well, photosynthesis solved the problem. Uses sunlight as an energy source rather than existing inorganic molecules. And the cyanobacteria were the organisms in which photosynthesis evolved, and they became extremely abundant by 2.5 million years ago, or 2.5 billion years ago or so. Very abundant. The oceans were filled with cyanobacteria. And because they were so abundant, and you know from the Bio 93 class where you've learned a little bit about photosynthesis, that photosynthesis operates by the transfer of an electron to a water molecule and splitting the water molecule, and oxygen is evolved as part of the photosynthetic reaction. So these things are all giving off oxygen now. And that free oxygen gradually accumulated in the atmosphere. That's why we have an oxygenated atmosphere. Now, that was a very, very big event in the history of the evolution of life on Earth. And the next big challenge was to be able to adapt to this aerobic environment. Organisms that had previously been anaerobic had to evolve and adapt to a very large change in the physical environment. So aerobic respiration evolved. It evolved in again, bacteria. Now, this is long before multicellular organisms. Things like alpha proteobacteria. We'll see later a phylogeny of the different bacteria, but things like alpha proteobacteria evolved the ability to actually use oxygen as an electron acceptor, an electron transport, which turns out to be very much more efficient compared to anaerobic metabolism because there's a bigger energy potential associated with using oxygen as an electron acceptor. So you could do a lot more. And in fact, that step, evolving aerobic metabolism, again, in things like alpha proteobacteria, was a key element in ultimately life being able to evolve multicellularity, because you really needed greater energetic efficiency for these multicellular systems to evolve. So this is one of the key steps, moving from using an organic or an inorganic molecule as an electron acceptor to oxygen as an electron acceptor, as oxygen became much more abundant, because the energy potential is a lot higher with oxygen. So it's a good test question. Energy potential is higher with oxygen, okay? So what happened next? Now I'm getting ahead of myself, but I'm on a roll here. What happened next? Well, you got anaerobic metabolism in a few bacteria, and then later, the beginnings of the evolution of the eukaryotic lineage has begun to appear, and at some point, those aerobic bacteria associate with an anaerobic eukaryotic cell, and that's where the mitochondrion in your cells comes from. It comes from an alpha proteobacterial lineage and not from a eukaryotic lineage. So you're a hybrid. You've got part of your genetic material comes from a bacterial lineage, and that's the part that is fundamentally important for oxidative phosphorylation, for the energy, the electron transport energy system that makes ATP in your cells has a bacterial origin. Well, there's another place where bacteria play a crucial role in the ecosystem of the world, and that's in the nitrogen cycle. All organisms require nitrogen to live. There's half a dozen key elements that make up most of the molecules that we're constructed of. Proteins are, carbon and hydrogen and nitrogen. They're amino acids. They've got nitrogen is a key part of all proteins, and yet it turns out that one of the weak links in the sort of system that we live on is the availability of nitrogen in a form that we can utilize. And you say, wow, why should that be? Because, like 70% of the atmosphere is gaseous nitrogen. It's into, we're awash in nitrogen, but we can't use it. We have no biochemical way to take into and use it in our metabolic processes. So it's just a gas that we're living in that doesn't contribute anything to us. But bacteria can do that trick, and Archaea. Bacteria and Archaea are able to fix nitrogen into ammonia and nitrate, into forms that can then be incorporated into protein synthesis and the construction of amino acids. Okay? So bacteria are hugely important to the nitrogen economy of the world. And if they went away, we'd be in a terrible fix. Because we would not, well, actually now we know how to fix nitrogen synthetically. So the reason we're using an overabundance of nitrogen in things like fertilizing crops is because during World War II, a lot of explosives are made with nitrogen compounds, nitrates and so forth. And so during World War I, there was a shortage of nitrate materials in Germany, particularly, and some German scientists invented a process that used high inputs of energy to make nitrates for explosives. So now we use that process and the sort of improvements on it as a way of making fertilizers for plants in agriculture, and explosives, of course. But before that, we were completely dependent on bacteria and Archaea as the ultimate source for nitrogen in a process which is known as nitrogen fixation. And only a few lineages of bacteria and Archaea can do it. If they disappeared, the rest of the world of life would be threatened. So certain aquatic species of cyanobacteria also fixed nitrogen. You can see these beautiful nodules. Well, we'll see a picture of a cyanobacterium in a little bit. And then on the land, there's some soil bacteria that fix nitrogen. But the most important source, particularly for plants and ultimately for us because we consume plants, are rhizobia. This is a group of bacteria which actually live in a very close symbiotic association with plants. And this is a plant root, and you see all those little circular nodules. Those are nodules that have been created by a colonization of rhizobia in the roots of the plant. And the plant actually provides carbon, sugars in effect for the growth of the bacteria. The bacteria fixes nitrogen and contributes that to the plant's metabolic needs. So this is a wonderful example of a symbiotic association, and it's one of the key places where fixed nitrogen gets into the ecosystem. We come along later, eat the plants, and get the nitrogen that we need from eating the plants. But that's nitrogen fixation. Very key part of the sort of global economy of the Earth. We would be in deep trouble without nitrogen-fixing bacteria. Okay, so now we're going to talk a little bit about how do biologists study bacteria and archaea. How do we actually go about studying these organisms? Well, they're not, we've been studying bacteria. By the way, one thing I meant to say about Koch that I forgot, and that is that he is also the person who essentially proved the germ theory of infection. Very, very important figure in the history of science. But so how do we study them? The reason that Koch's name came back to mind is, of course, one way is we use cultures. That was the classic way. We try to see if we can grow them. Well, if you want to grow a bacterium, how do you go about it? One of the things you've got to decide is, well, what do they like to eat? Okay? What is it that they need to eat? So a lot of work was done defining what are called the minimal media, the minimal requirements for various kinds of microorganisms to actually live, and then to create media that they can subsist on and see if you can then grow them as pure cultures. And it used to be, back in the old days, let's suppose that you were suspected of having something bad like tuberculosis. Well, what they had to do was take a sample, culture it, see if something that looked like the tuberculum bacteria grew out of that culture, and tell you 10 days later that, yeah, I'm sorry, but you've got tuberculosis. Well, now we can do it overnight. We have much more powerful approaches to studying bacteria, but the original ones were just culturing. And so these enrichment cultures are the first step, and they're a powerful step still. And I want to discuss just a bit an example which is in your book, which is a wonderful example of using enrichment cultures to ask a surprising question about bacterial life. One of the things that was discovered beginning in the early 1990s, I remember the first time I heard about this was about 1993 or so, 1992, was that there are bacteria that are found when you bore into the earth deep, like, 4,000 or 5,000 meters, 15,000 feet, you're boring way down, you find, and you take out these core samples, which are pure material, and you find that there are hints of microbial activity in them. And that's incredible. What in the world is life doing way down deep under the earth? And these, by the way, the temperatures in these environments are high because they're far enough down where the temperature is higher. It's anywhere from, say, 45 to roughly 80 degrees centigrade, very high temperatures, extreme environment, extremophile. How do they subsist? There isn't other carbon material that they can use to digest and get energy from, so how is their energy metabolism working? Well, one of the nice experiment that was done early on to prove that was an experiment that is shown in your book. This slide comes directly from your book. Material was taken from one of these deep bores. It's material from a bore in Virginia. It's about more than a mile deep under the earth. And the question was, the first thing they had to do is get some idea about what are the nutritive requirements. And so based on the physical environment, the hypothesis emerged that, well, maybe they're actually using hydrogen, H2, hydrogen, as an electron donor for electron transport. And maybe they're using ferric oxide as an electron acceptor. So if that's the hypothesis, so you take some of this material, grow it in a flask like this, and if that hypothesis is correct, what you'll observe is that the ferric should be oxidized to another material called magnetite, Fe2O4, and you should find magnetite in the resulting culture, building up over time. So they did the experiment. You put the sample in, give it what you think are the minimal things it requires, hydrogen gas and ferric, and you then raise it, you grow it at a temperature which is high, which is like the temperature that presumably it subsists at under the earth, and by golly, after a week or so, you can detect magnetite. Amazing, proving that this is that hypothesis, or at least consistent with the hypothesis that these organisms are actually using hydrogen gas as an electron donor, ferric as an electron acceptor, empowering their metabolism using these inorganic molecules deep down under the earth. Well, they examined the resulting cultures with electron microscopy, scanning electron microscopy, and you can see the pictures of the bacteria. These things are growing more than a mile under the earth. It's an amazing story. So that's a wonderful example of the use of culture techniques. Now, today, there's a different approach that's mostly being used, and that's what's called direct sequencing. And so here's the idea. There's some molecules that are common to all life. A good example is the ribosomal RNA molecules, because that's where protein synthesis occurs, and all organisms use ribosomal RNA molecules for protein synthesis. So you take the small subunit of the ribosomal RNA. It's the same principle that, remember, I've talked about Carl Woese, same idea, and you sequence it from a lot of different organisms, a lot of different bacteria, so that you build up some picture of the bacterial tree. And then you go out into the environment. And you can't culture most bacteria. Remember I said that there are about 5,000 species have been described, but that we think there are millions, but we can't culture most of them. So you go out into the environment, take a sample of the environment, and you simply make DNA from that sample. Okay? You just grind it up. You'd be surprised how much living stuff is in any kind of sample. Seawater, soil, whatever. You can recover DNA from it. But what you recover, this DNA is going to be a pool of DNA contributed by all the organisms that were in that sample, in the soil sample or the water sample, the ocean sample, whatever. So the next step is that you use this PCR method to amplify ribosomal RNA molecules, and you sequence them. You see how many different sequences you get. And you can associate each sequence with the things that are most related to it on the tree of life, and you can say, oh, well, this one belongs over here. It's something we never knew about, but we know where it should go on the tree of life because it's closest in sequence to some other things that we have already looked at. So in that way, using this direct sequencing approach, two of the major lineages of Archaea have been discovered. They're given here. And there's a really famous scientist, colorful guy named Craig Ventnor. Ventnor, who's actually shown, that's the bald guy is Craig Ventnor. He's about my age. Vietnam War veteran, a poor student. After he got out of the service, he got his act together and went to UCSD, ended up with a Ph.D. in biochemistry. And then he just didn't stop. He became a very imaginative and effective researcher at the National Institutes of Health, and among other things, he created several companies. There were two groups that first sequenced the human genome. Craig Ventnor's group, which was a private effort, was one, and the other was an NIH-led effort. And anyway, Ventnor's a very creative guy, and he's made a lot of money in this. He created a very successful company called Cellular that did direct sequencing of a lot of different organisms. And so he bought himself a nice boat called the Sorcerer II, and he's traveled around the world, and it's shown in this panel in the lower left, making samples of ocean samples and then sequencing these things by the methods that I mentioned to identify new forms of life that we can't culture and hadn't seen before by this methods that I've already described. So, this is one of the great periods of biological discovery again. We're actually in this era, just in the last decade or so, discovering more different forms of life than any time since the 19th century. So that's two of the ways that we have to study and identify bacteria. Now, I want to move on just for the last couple of minutes and introduce a topic on metabolic diversity, because the other thing about bacteria and archaea that's important to us is that they have a lot of diverse ways of driving their metabolism. That's why they exist in so many different environments, why they're so adaptive and successful. They have many different ways of approaching the problem of living, many more ways than we do as eukaryotes. And so all organisms acquire chemical energy and use it in the form of ATP. Okay? ATP is one of those fundamental universal molecules that is the energy source. It's sort of the gas tank for all organisms. But bacteria and archaea have a lot of different ways to drive ATP production. They can use light, that is photosynthesis. They can use organic molecules. That's what we use. We use organic molecules. We eat things, digest the organic molecules, convert that into ATP. But they can also use inorganic molecules as sources of ATP. We saw an example already when we talked about the bacteria that are deep underground. So there's a huge amount of metabolic diversity for bacteria, and this table from your book just summarizes the 6 different ways that bacteria and archaea can obtain energy and make carbon-carbon bonds. And we'll pick up here on Monday.